We're going to finish up glycolysis. We're just a couple of enzymes away from the end of glycolysis. And then we're going to talk about what happens depending on the oxygen state. And then we're going to go backwards and do gluconeogenesis. So we should be able to get through all of that today. We're also going to hear a silly little song that really probably won't help you learn glycolysis, but maybe it'll give you something to chuckle about. So where did we leave off in glycolysis? So I think we ended last Thursday with making 2-phosphoglycerate. So remember we talked about isomerizing from 3-phosphoglycerate to 2-phosphoglycerate. So what was the intermediate for that reaction? BPG, 2,3-bisphosphoglycerate. So remember that was the enzyme that has a phosphorylated histidine residue, is the mutase. And so we ended up with this intermediate 2,3-BPG, which reminded us of hemoglobin. And the whole thing about hemoglobin is oxygen transport and delivery, and we need oxygen in glycolysis. Well, we don't really need it in glycolysis, but ultimately we need it in respiration. And so that's how those two features of biochemistry that we learned independently are interconnected. And so hopefully we'll see more of those connections for the rest of the semester. And so the next enzyme in glycolysis, so we are almost there. This is the 9th enzyme of glycolysis, is enolase. And so if you just had to guess, what class of enzyme would enolase be? Lyase. Okay, so lyase, because we're making a double bond. Okay, so in one direction we're adding across a double bond. In the other direction, so in the glycolysis direction, we're removing water to make a double bond. Okay, so it's a condensation reaction. Not really. Is it a condensation reaction? We're removing water, but it's not condensation like peptide bond formation. So, but we're definitely removing water. And so, what's the big deal about phosphoenolpyruvate? Well, so we're going to see that now pyruvate kinase, which is the next enzyme, will take us to yes. So we have phosphoenolpyruvate. Sorry. Now we can dump that phosphate group from phosphoenolpyruvate to a molecule of ADP for our second ATP production reaction. Okay. So this is basically the second payoff step. Okay, and this kind of production of ATP is called substrate level phosphorylation. Okay, because we're going to see in the electron transport chain and in the mitochondria how we make ATP through the ATP synthase enzyme, but this is a different way of making ATP. So we've now seen 2 kinase reactions. So remember, we originally learned that kinase reactions use a molecule of ATP to phosphorylate something, and so that's kind of the universal way we tend to think of kinases. So both of these kinase reactions in the payoff phase of glycolysis are actually named for the other direction. Okay, so both of those reactions actually make ATP because the precursor is actually at a higher energy level than ATP is. Okay, so remember we saw 1,3-bisphosphoglycerate, how it can donate that phosphate group to ADP, and that's energetically favorable because that's a higher energy bond than the ATP anhydride bonds. Same thing for phosphoenolpyruvate. So now that phosphate group, cleavage of that phosphate group is favorable even as you're making ATP. Okay, so that's called substrate level of phosphorylation. So ADP is a substrate for the reaction. We're just swapping out that phosphate to make a molecule of ATP, which is kind of the point of glycolysis, one of the points of glycolysis in the first place. So we're using energy, we're using the fuel of glucose to turn it into the fuel of ATP. Does that make sense? Okay. What else did I want to say about pyruvate and ATP? Maybe something. Maybe it'll come back to me. I can't remember. Anyway, 2 kinase reactions. Yes.
Is one thing BPG also subject level phosphorylation?
Yes. Yeah. So both of those reactions, and so What's the name of that one? We'll come to that overview. So somebody tell me the name of the. There we go. Yes. So phosphoglycerate mutase. Or sorry, phosphoglycerate kinase is also substrate level phosphorylation. Thank you. Okay, so we have 2 steps now in the payoff phase that make ATP for us in ways that are energetically favorable. Okay, so we're getting the payoff. And so what's the big deal? So why is making pyruvate so favorable? So if cleaving off that phosphate group from phosphoenolpyruvate is energetically more favorable than cleaving the ATP, cleaving off the gamma phosphate of ATP, why is that so? And so it's because we can draw other tautomers, and so we have a more stable product. So the pyruvate is more stable because we can draw different tautomeric forms of it. Okay, and so when we draw, so remember if we had the phosphate group here, right? So if that was, the phosphate group, and we cleaved it off, it's kind of like it's written as the enol form, right? But once that phosphate group comes off, then it can tautomerize. Okay, and so then that's going to be energetically more favorable. And so now we have pyruvate. Ta-da! We've made it all the way through glycolysis. So if I could have talked faster on Thursday, we would have done all glycolysis in one lecture, but that might not have been a good idea. So now as you think about all these 10 reactions, so even though they seemed daunting, hopefully, when you first looked at them, now they're not quite so daunting. Okay, so we've seen kinases acting as kinases should, in our opinion, right? So using a molecule of ATP to phosphorylate something that then is going to drive that next reaction forward. And then in the payoff phase, we also see 2 kinases that produce ATP for us. Okay, so all of a sudden there's 4 kinase enzymes. You know how kinases work, and that's 4 out of 10, right? And so those are pretty straightforward. Oh, there's another kinase. So 1, 2, 3, 4. I ticked the wrong one there. And so we have a couple of isomerases and a mutase, right? So now that's 7 reactions that you've worked on. Aldolase, we said, oh, that mechanism is yucky, messy. We're not going to learn it. Okay, we didn't talk about the enolase mechanism, and we learned the dehydrogenase mechanism. Okay, so you have lots of kinases. You have a couple of isomerases, which we practiced, and you have the dehydrogenase, which we're going to see 2 more examples of today. So you'll have those under your belt by the end of today. So hopefully that makes you feel better about drawing the molecules and learning the mechanisms. So if we look at glycolysis overall and the energetics of it, what do you see about both the standard state free energy changes and the real free energy changes? What are some things that stand out? Right. Okay, so we talked about near equilibrium metabolic reactions and metabolically irreversible. Katie?
They're not near equilibrium, they're negative, so it would be favorable.
Right. Okay. So we hope, we're going to do a lot of glycolysis in a lot of cells, so we hope that the ones that are not near equilibrium are favorable in the glycolysis direction, right? And we're going to see, oddly enough, that those reactions that are metabolically irreversible in glycolysis, we're going to have to deal with in gluconeogenesis to get gluconeogenesis to be favorable overall too, right? And so the ones that have a slight positive, fortunately, when we have substrates building up from the previous reaction, they're close to equilibrium, so now the abundance of substrates will drive those reactions forward. Okay, so then our real delta G values give us near equilibrium values. So how many of those reactions are near equilibrium? 7 out of 10. So we said the majority of metabolic reactions are near equilibrium. So that makes sense. I mean, that fits with what we said. And then there are 3 that we would say are metabolically irreversible. Okay, and so we're going to talk about, so what did we say about metabolically irreversible reactions in metabolism? They're often control points. They're often highly regulated. So we're not going to talk about the regulation today. We'll talk about it next week, right before the exam. Not going to be on the exam. But we'll talk about how those 3 enzymes in particular have many layers of regulation because it's so important So because they're so energetically favorable, we don't want to go down that path unless we really want to do glycolysis. Okay, and it makes sense that the first step of glycolysis is one of those metabolically irreversible steps. So once you have glucose and the right regulatory conditions, we're going to enter glycolysis and then pretty much keep going. It's kind of, you're jumping into the rapids and going downhill. But we're going to see those metabolically irreversible reactions are both controlled and regulated, and they're the ones that we need to deal with for gluconeogenesis. How are we going to get up the rapids if it's so favorable to go down the rapids? Okay, so that's just another graphical way of seeing those free energy changes. So here, here's the whitewater rapids that we're going down if we have an abundance of glucose as a metabolic fuel. Okay, so again, if we're going to have glucose as a fuel, where does it come from? So it could come from just, sugar in your Frappuccino, right? And so that would be a good glucose or probably fructose sucrose store. But if you're, if you don't have high blood sugar, where would you want to get that glucose from? You're going to have to get it from glycogen. That's stored in your muscle or your liver, and then how do we process that? So how do we break that down? So remember we talked about the reducing end and the non-reducing ends, multiple non-reducing ends of glycogen. So if we have, the core there, and then you have all these branch points, right, of glycogen. Now you have all these non-reducing ends where you can go in and clip off enzymatically all those glucose monomers to then feed into glycolysis. And so we're going to talk later about the enzymes that both make glycogen and break down glycogen, how they work and how they're regulated. But I'll show you now that the enzyme is called glycogen phosphorylase. Okay, and so the enzyme that breaks down glycogen, so it's kind of like hydrolysis, but not, because it uses a phosphate molecule to actually break the bond. Okay, so where did we see that before? Do you remember a phosphorylasis reaction? So it was a mechanism that we did last Thursday. An enzyme that used covalent catalysis, and then to cleave off the product, it used a molecule of inorganic phosphate. Right, so glyceraldehyde 3-phosphate dehydrogenase. So remember, it uses covalent catalysis, so there's a cysteine residue that attacks the substrate. We have reduction reaction with NADH. And then to cleave off the product, we don't use a molecule of water, we use a molecule of phosphate. Same idea here. So we use that phosphate molecule to cleave, so it attacks there and cleaves that bond. And now what's our substrate? So we have glucose 1-phosphate. Is that what we need in glycolysis? So is that one of the intermediates of glycolysis? No. So remember, we learned glycolysis starting with what? Glucose. What's the next metabolite in the pathway? Glucose 6-phosphate. Okay, so wouldn't it be easier if we made glucose 6-phosphate? Well, it might be, except we have to cleave off at that one position. So what we end up with is glucose 1-phosphate. So do you think we move the phosphate group? Okay, so yes, we do, in fact. Okay, so if we start glycolysis from glycogen, we do phosphorylasis to make glucose 1-phosphate, which then isomerizes to glucose 6-phosphate and then continues with the rest of the reactions of glycolysis. Do not memorize that slide. Okay, so this is just saying that even though we kind of narrow-mindedly learn the glycolysis pathway starting with glucose, we don't just consume glucose. Okay, so we might be consuming sucrose. So where do we find sucrose? Table sugar, right? So sucrose is a disaccharide of glucose and fructose. So if we process the sucrose, we have a molecule of glucose. What do we do with the leftover fructose? Well, we have another enzyme that will feed that in. Okay, so even though we learned from glucose to glucose 6-phosphate all the way down, our body is very good at adapting to whatever sweet thing we give it. Okay, so we have a whole lot of other things. So galactose, mannose, fructose, There's probably some lactose. There we go. Okay, so any of the sugars in any of our foods, we have enzymes that will break them down. Okay, so this one sometimes is a problem. So my mother, the pediatrician, used to always say, you know, all these campaigns to get people to drink milk, you know, have the milk mustache, drink more milk. No other mammal drinks milk after weaning. It's just a scam of the dairy industry. She's like, you don't need that much milk. So what's the evidence that adult humans don't really need as much milk as the dairy industry tells us we need? So we tend to lose lactase as we grow up. Okay, so that enzyme lactase, really, you know, we don't need it as much as we grow up. And so many adults, older adults, have problems digesting milk sugar. Okay, so why does the dairy industry try to sell us milk? Or why should we drink milk, maybe? What are the good features? Calcium, vitamin D. So there's lots of good things in milk. Some of that is added in, and really we could get it from other sources. Okay, so if we had green leafy vegetables, we'd have plenty of vitamin D. If we ate, where else do we get our calcium from? Lots of other places. Okay, so many cultures don't drink milk as much as Americans drink milk. There's nothing better with an Oreo cookie, but you really don't need that much milk. Okay, that's my soapbox for today. Okay, so that was just to say we have lots of sugars in our diet. For the most part, we can deal with them, and then they all ultimately feed into glycolysis. Okay, so again, we tend to learn these enzymes as isolated molecules. We can certainly study the structure of any of those. And so we kind of tend to think, oh, you have a substrate being made into a product. Oh, then the next enzyme downstream, you have that as the substrate becoming a product. It's actually very inefficient if you think of each of those enzymes as isolated proteins doing their chemistry. Because what happens if a product of one enzyme diffuses away from the enzyme that it's a product of, it has to find the next enzyme in the pathway, right? And so there could be degradation of that product before it ever gets to the next enzyme. So it turns out that many enzymes that are involved in metabolic pathways are actually hanging out together in the cell. Okay, and so this is called substrate channeling. Okay, and so instead of having the product, you know, one of the metabolic intermediates diffuse away and possibly become degraded or find another enzyme that isn't, you know, what's next in the pathway, if those enzymes are near each other, it's kind of like a handoff. So the product of one reaction gets handed off right away to the enzyme that's downstream, and you don't have diffusion away of that intermediate. Okay, that sometimes, you know, so sometimes those are not obvious to see, and we can't capture those complexes because when you break open the cells, they kind of go all over the place and get scattered. But in reality, they kind of hang out together to do their work together. Okay, so we've made it to pyruvate, right? So we just did all that. And so does anyone have questions about any of those steps? Yes, Jessica. Mm-hmm. For which ones? No. Do not memorize those. No. So the glycogen phosphorylase we're going to come back to and study more in detail because that's one of the enzymes that's actually very highly regulated. So we'll come back to regulation of that. But all the other sugars feeding in, you don't need to know those. Okay, so then what happens to pyruvate? So what happens to pyruvate depends on what kind of organism you are and what are the oxygen conditions that you're faced with. Okay, and so if you're a mammal over here, right? So you might have abundant oxygen supply, and then we're going to talk about respiration a little bit later. But what happens if you don't have enough oxygen and can't do respiration? So what kind of situation would that be? So what if you're suboptimal oxygen levels? So if you're sprinting, right? So running away from the bear, you're probably going anaerobic, right? Or just plain sprinting for the fun of it, right? So your muscles tend to be undergoing anaerobic metabolism. Do you know what other cells produce a lot of lactate? What other cells don't have enough oxygen? Well, so muscles under like high energy demand. So red blood cells, okay, are also oxygen deficient, or at least don't have a surplus to do respiration. Or if you're a microorganism that undergoes fermentation. Okay, so we'll talk about each one of those and what the product, where pyruvate goes in each one of those phases. Okay, so lactate dehydrogenase. So again, under low oxygen conditions. So again, if you're a big animal like us, it's hard to get enough oxygen to your muscles if you're sprinting fast. So high muscle demand. Our hearts kind of can't keep up with getting the oxygen to all the distant muscles. And so we have this challenging situation that we can't take pyruvate all the way through acetyl-CoA to respiration in the mitochondria. And so we have this kind of temporary stopgap measure. Okay, so pyruvate is oxidized to lactate and with the enzyme lactate dehydrogenase. So as soon as you see lactate dehydrogenase, what should you think? NADH. Okay, so dehydrogenase, you should automatically think it's an oxidative reductase, and NADH is the cofactor. So again, what reaction did we do last week? It was a dehydrogenase. Glyceraldehyde 3-phosphate dehydrogenase, right? Okay, same deal. Same deal. It's a dehydrogenase that uses NADH. Okay, so lactate dehydrogenase. Again, we're going to use NADH. That's an ugly NADH. Okay, so if that's the business end of NADH, and there we have pyruvate. So again, you're going to learn these structures. So this is a reduction of pyruvate. Did I say oxidation to lactate? It's a reduction of pyruvate to lactate. Sorry about that. And so if it's a redox reaction using NADH, how are the electrons transferred? In what form? As a hydride. Okay, so dehydrogenases use NADH and undergo hydride transfer reactions. Okay, so now that hydride is going to get transferred to the pyruvate. So we're going to have a proton floating around, so that's going to grab that proton, right, to make L-lactate. So this is the reduced form of pyruvate. So NADH gets oxidized. Yes, sir.
Is the hydride leaving, is that stabilized by resonance? Is that why?
The hydride leaving.
The
Yes, yeah. Sorry. So now we have NAD plus. Yep. Okay, so what does that get us? So why does that help the situation? So what would happen if we were exercising vigorously, so sprinting? So how much ATP do we get from our muscles from glycolysis? Just a little bit, a little bit. What would happen if we didn't get rid of the pyruvate, but also didn't have enough oxygen to continue on to the citric acid cycle and respiration in the mitochondria? It would stop. Okay, so if we have a buildup of pyruvate, hypothetically, we could go backwards into gluconeogenesis. That's not what we want to do, right? We want to, if we're sprinting, we don't want to kind of fall apart halfway down the 100-meter dash, right? Or if we're running away from the bear, we would hate for the bear to catch up with us. So you want to keep glycolysis going. So this lactate dehydrogenase reaction allows glycolysis to continue even though there's not oxygen to ultimately be the electron acceptor. Okay, so when we get to mitochondria and electron transfer reactions, oxygen, so O2, gaseous oxygen is the ultimate electron acceptor from all these reduced cofactors that we're making. But if we don't have oxygen, we have both a buildup of NADH and a buildup of pyruvate, and that's not ideal. Okay, so what do we need? So where did this NADH maybe come from? So what was the other dehydrogenase reaction in glycolysis? The one we just did? So glyceraldehyde 3-phosphate dehydrogenase, right? So if in glyceraldehyde 3-phosphate dehydrogenase we made NADH, right? So which half of the redox pair do we need for glycolysis to continue? We need NAD+. Okay, so glycolysis needs NAD+. So if we want to keep glycolysis going, but we've built up our NADH, we need to interconvert them. Okay, so in order to keep going in glycolysis, so we probably have a sufficient amount of glucose, but if we're stuck in pyruvate, we can't keep making more pyruvate. We can't keep making that little bit of ATP that we get from glycolysis. Does that make sense?
So you need the NAD plus, not
So,
necessarily lactate.
right. So the lactate is not going to help us for anything, except in Le Chatelier's principle, right? So if we have this metabolic pathway, if we pull out the pyruvate, so if we deplete the system of pyruvate, we can keep going forward in glycolysis. So we've both depleted the pyruvate and we've regenerated NAD+ to feed back into glycolysis. Does that help? What, is everybody okay with that? We'll keep talking around that issue for a little while. Okay, there. That's what I just said. So if that helps a little bit more. Okay, so in glycolysis, glucose going to pyruvate. So the step that needs that, right, is making, so glucose, glyceraldehyde 3-phosphate dehydrogenase uses NAD+ to make NADH, to make pyruvate ultimately. So now we have this buildup of NADH. We can recycle the NADH back to NAD+ in lactate dehydrogenase and deplete the pyruvate so that we can keep going forward in glycolysis. Okay, and so this happens in muscle cells. Okay, so it allows glycolysis to go forward, but now we have this buildup of lactate. So what happens to the lactate? So it actually, in the liver, gets converted back to pyruvate. Okay, and so this happens in the liver. Okay, so in the muscle cells, we have this buildup of lactate. It gets transported in the bloodstream to the liver, and then in the liver, we get the lactate converted back to pyruvate in the presence of oxygen, so under resting conditions. Okay, and so this interconversion, so kind of all these interconversions, pyruvate to lactate, lactate back to pyruvate in different tissues, is what's called the Cori cycle. You guys learned that, those of you who've had cell biology? Do you learn about the Cori cycle? Okay, so the
Sorry, can you just repeat the definition?
okay, so the Cori cycle, well, so I think, like in a narrow sense, the Cori cycle is pyruvate to lactate in the muscle, and then lactate back to pyruvate in the liver. Okay, so and all the transport happens through the blood, obviously. And so the Cori cycle is named after Gertrude Cori, and golly, I always remember the wives' names, not the husbands' names. I know that's terrible. And her husband, Carl and Gertie Cori. Okay, so Carl and Gertie, their pictures are in your textbook. Gertrude, Gertie. Okay, so they were partners in the lab, husband and wife partners in the research lab, who were very savvy about how they published their papers. So they each had, so they worked together their whole scientific careers, but they each had they each had their own projects as well as shared projects. And so sometimes she would be the primary author on a paper, and sometimes he would be. So when the time came to award the Nobel Prizes, they couldn't deny that she had done an equal amount of work, because he wasn't always the communicating author on the paper. That's not why they did it, I don't think, because nobody ever plans to get a Nobel Prize except for Jim Watson. But they were clearly both independent and a team. And so they were working, I think, at University of Washington at St. Louis, studying the enzymes of sugar metabolism. And so they figured out a lot of how these enzymes worked in glycolysis, gluconeogenesis, and then the interconversion. So they won the Nobel Prize in the 60s, maybe, I think. Anyway, very smart researchers. Yeah, so requires resting phase, including high O2. Okay, so those of you who are runners, can you kind of apply this to how it works when you sprint? So then when you stop, you kind of go, so you're getting more oxygen to your tissue, and then the lactate in your muscle gets transported through your bloodstream to the liver. Now there's sufficient oxygen levels that it can get recycled. Carter, you have a question?
Yeah, is the Fourier cycle then? Is the point of it to reconvert this catalyst so that it can be used again?
So it's not really to the catalyst is the enzyme, so there's always plenty of the enzyme there. The purpose is to, now that you have high oxygen levels, we're going to see the next step where pyruvate goes. So after we talk about gluconeogenesis, pyruvate then can get made into acetyl-CoA to enter the citric acid cycle. And then ultimately, all those reduced cofactors can enter the electron transport chain in the mitochondria to make more ATP with oxygen as the electron acceptor. So basically, in the absence of oxygen, everything comes to a stop. And so we can't get all the energy out of the pyruvate that we know is there chemically. So the point of the Cori cycle is, so you're not going to enter the citric acid cycle from lactate, you're going to enter it from pyruvate. So you have to get back to pyruvate to then enter the citric acid cycle. Yep, so it's really just coming back to where you kind of put things on hold, and now you have enough oxygen, you can keep going in the process. Yes?
So is lactic acid just the proteinated form of lactate?
Yes. Yep. Yeah. So lactic acid is just, so lactate is the carboxylate. Lactic acid would just be that. Yep. Okay. So before we talk about beer, we're going to, so your break is going to be listening to this silly little song. If you need to get up and stretch, you can. But in the meantime, we don't really want to end it. Yes, keep and audio and okay, so my favorite silly song about science? Maybe? Oh, come on. Well, maybe this isn't going to work. Okay, you're going to have to listen very closely.
You're sweeter than a woman's kiss, 'cause I need you for glycolysis.
Okay, listen very closely. We're going to start again. You can read the lyrics. I'm going to try again.
Oh, sugar, sugar.
There we go.
You are my favorite.
Wait. Try again. Okay. Full screen. You're asking a lot. Catch your tune.
You are my favorite. You are my favorite. From the blood-borne substrate. Monosaccharide sugar. You're sweeter than a woman's kiss, 'cause I need you for glycolysis. I just can't believe the way my muscles take you in for you. All it takes is a little bit of influence.
So GLUT4 is the glucose transporter.
Oh, sugar, you help me make ATP when my predators are chasing me. Oh, glucose, you're an aldehyde sugar, and you're sweeter than a woman's kiss, 'cause I need you for glycolysis. I just can't believe the way my muscles break you down. A few more seconds and I'll be rigor mortis style. You're sweet and turning sour, baby. I'm losing all my power, baby.
Make your muscles ache.
No, 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 I'm swimming in lactate, baby. Oh yeah, I'm swimming in lactate, baby. Now I'm drowning in lactate, baby.
I'm gonna make your muscles ache.
No, 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 I'm swimming in lactate, baby. Oh, glucose. Oh, sugar, sugar. I used you up and you left me flat. Now I'll have to get my kick down flat. Oh, glucose. Sugar, sugar. The
Almost done.
honeymoon is over now. The honeymoon is over now.
Catch a little tune. Wow, that's really silly. Yep. So you know the I guess not urban myth, because it's not really an urban myth, is that the burn in your muscles is lactic acid buildup. Not entirely true, apparently. I'm not sure why it's not true, because it makes sense to me. But apparently it's not entirely true. But that's why it says, I'm going to make your muscles ache. I'm swimming in lactate. Oh, no. Stop. Cancel. Come on. Okay. Oh, I'm just going to do that. Go to slide. Okay. Okay, so how many of you have ever had a tour of the brewery at Foothills? Have any of you ever been to Foothills? Okay, good. If you're well, I actually don't have to go. They have very good food too. But if you're ever there when it's kind of a slow time, I don't know, in between lunch and dinner, late afternoon, ask for a brewery tour, because they will give you a tour of the vats and kind of talk about the enzymology of beer making to some extent. Especially if Jamie is there, the brewmaster. He's really good at kind of talking about the science of beer making. And so, if you're a yeast, you can make alcohol, right? And so he'll show you there. You know, he'll let you smell the hops. They're kind of cool, and the barley, and they have this really cool whole plumbing system, auger system set up to move things from one area to the other. So they'll cook the barley a little bit. So that's going to break down the cell walls to make this malt, and then gradually more cooking, but not too hot. So if you think about an enzyme lab, you don't want to bake the enzyme. So you have to break down some of the parts of the cell without actually denaturing the protein at high temperatures. Okay, so once you have kind of this mixture of simple sugars, then you want to add the yeast. Okay, and so what's the difference between wine making and beer making? Okay, winemaking uses grapes. Okay, so did you know that when you make wine, you don't have to add yeast? So why don't you have to add yeast when you make wine, but you have to add yeast when you make beer? In wine? No. Okay, what makes them ferment? Okay, so it turns out that grapes actually have, and grapevines have microorganisms, yeast and other microorganisms, fermenting microorganisms that live on them naturally. Okay, so that's why fruits will ferment naturally, because they already have stuff living on them. Okay, and so the different kinds of wine that's made around the world is not just the varieties of grapes, but the varieties of microorganisms that kind of come along with those grapes. So when you take a rootstock from France and bring it to the U.S., you know, you may have the right grapes, but unless you have the right kind of microorganism environment, you might not end up with the same taste of wine. Isn't that cool? So the grape juice will ferment naturally because it kind of brings along its own microorganisms, but the barley and malt, you have to add yeast in. And again, the kind of yeast you add in is going to change the fermentation process and change the flavor of the beer. Okay, so, what's that? Is barley a weed? Oh, it's a grain. It's a grain. Yeah, it's not exactly wheat, but it's a grain. So if anyone here has gluten problems, celiac or other gluten sensitivities, beer should not be your drink of choice. Okay, wine is fine, but beer is not. Okay, so I have a friend who was diagnosed probably when she was 30 with celiac disease, so British heritage, not at all surprising that she would be diagnosed with this, but she, you know, would drink beer in college, grad school, and be like, why does it make me so sick? I feel so lousy, you know, so she thought she just couldn't drink. And then when she found out she had celiac disease, she's like, oh, it's not the alcohol, it's the grain. It's the wheat and barley and the gluten in that's the problem, not the alcohol. So wine's not a problem. Okay, so once you have all the simple sugars, now you add in the microorganism. So depending on whether you have oxygen present or not oxygen present will dictate the end products of the reactions. Okay, so if we have C6H12O6, you know, so glucose and other simple sugars, now we can feed into glycolysis, citric acid cycle, and electron transport chain, ultimately get all the way to CO2 and H2O. So that's complete metabolism of glucose all the way to its final end products with oxygen as the electron acceptor. However, in the absence of oxygen, now we're not going to fully metabolize to CO2 and water. We're going to metabolize to ethanol and CO2. Okay, so in beer making, for a while you kind of let the fermentation go in the presence of oxygen, and then you cap the whole thing, and then the oxygen is depleted as the yeast does their thing, and then at some point you go from converting the sugar all the way to CO2 and water to making ethanol. Okay, so you limit the access to oxygen, and the yeast do their thing and make ethanol. And so what are the two enzymes that are needed for that? So to get from, so we're just going to start with pyruvate, to get from pyruvate to ethanol, there are two enzymes that are used. And so the first is pyruvate decarboxylase. So we're going to flip off that CO2 group. So we're not going to do the mechanism for that. It's kind of ugly and messy. But I will point out the cofactor that's used. Okay, so the cofactor is called thiamine pyrophosphate. Okay, so and where might you have heard of thiamine before? Does that sound at all familiar? Okay, so it sounds like one of the bases, but it's actually, well, so this, I mean, so that's like thiamine, right? It looks kind of like thiamine a little bit. Does it sound like a vitamin? Yes. Okay, so remember we said early on in the semester, coenzymes are important, and in fact, often vitamins in our diet are precursors to coenzymes. Okay, so remember we said that the functional definition of a vitamin is something we need in our diet, and if we don't get it, there's some kind of defect that's fixed when we put it back into our diet. So the reason there's a defect is typically these are used to make coenzymes, and so then we can't do the enzymatic reaction because we don't have the coenzyme because we didn't have the vitamin. Okay, so thiamine pyrophosphate, all I want you to remember is that those are often involved in decarboxylation reactions. Okay, and that's a handy tip to remember if you're taking MCATs and other standardized exams. So there's lots of cofactors that we're going to learn, so it's often useful to think about what do those cofactors do in enzymatic reactions. So thiamine pyrophosphate is involved in decarboxylation reactions. Okay, so that's the first step. And then the second step, is yeast alcohol dehydrogenase. Okay, so in yeast alcohol dehydrogenase, we're going to reduce the aldehyde, okay, at that position by hydride transfer from alcohol dehydrogenase. So again, as soon as you see dehydrogenase, what should you think? NADH? hydride transfer. Okay? So if we have, so this is called, oops, acetaldehyde and ethanol. Okay, so if we have our NADH, so again, once you know one dehydrogenase mechanism, you know them all. So it's going to take me more time to draw the NADH than it's going to take to do the mechanism. Right, so it's going to be a hydride transfer. So a hydride transfer there. We're going to open that up. So yeast alcohol dehydrogenase, that's what YADH stands for, is our first metalloenzyme. Okay, and so we're going to move those electrons around like we did before. So the zinc, so what's the purpose of the zinc there? Right, stabilize the oxyanion. Okay, so remember when...
The oxyanion hole.
So it's kind of like the oxyanion hole of chymotrypsin. So remember when we make the oxyanion of the peptide bond, we have the backbone with its protons, its amide protons, stabilizing the oxyanion. Now we have something even more dramatic, which is a cation. Okay, and so that kind of makes it look like it's grabbing the zinc, which it's not doing. It's grabbing the proton, which is around there. So the zinc is going to stabilize the oxyanion, and then it's going to get, that oxyanion will get protonated. And then we have the NAD+. Okay? So again, once you've seen one dehydrogenase mechanism, you've seen it all. Hydride transfer using NAD plus NADH. Okay, and so that makes our ethanol for us. So do humans have, sorry, I should have asked that question. You want me to turn it back? So do humans have alcohol dehydrogenase? Yes, we do. Okay, or else we wouldn't be able to drink ethanol. Okay, so we don't make ethanol. Our liver alcohol dehydrogenase goes the other way. Okay, so yeast makes ethanol. We metabolize ethanol in the liver. Okay? So we metabolize ethanol. So can we metabolize methanol? We can, but it's not good for us. Okay, so the problem with liver alcohol dehydrogenase, so LADH, instead of yeast alcohol dehydrogenase, YADH, the problem with liver alcohol dehydrogenase is it's not as specific as we would hope. Okay, so sometimes we call those enzymes that are not as specific for a certain substrate, we call them promiscuous enzymes. So they're happy to take more than one substrate. And so the problem is the metabolite of ethanol, so aldehyde isn't so bad, or acetaldehyde isn't so bad for us. The metabolic byproduct of methanol or ethylene glycol is much worse for us. Okay, so yeast alcohol dehydrogenase sees methanol and says, yeah, close enough. Or it sees antifreeze, right, ethylene glycol, and says, yeah, close enough. And so the problem is not that the methanol is bad for us, it's that when our liver alcohol dehydrogenase metabolizes it, then the byproduct is toxic to our kidneys, to liver, all kinds of things. Yes?
Does that form oxalates?
So it forms, so if ethanol forms acetaldehyde, right? Formic acid, yes. Okay, so not so healthy. Okay, so you're making formic acid, which is then going to freak everything out. Yes?
I thought antifreeze was toxic because you go to sleep or something. Is that completely wrong? Or is it?
I believe it's completely wrong. Yes. I mean, it is toxic. It is toxic, but I don't.
But how long does it take? Is it a matter of?
Well, so it depends how big you are. So the problem with antifreeze tends to be small animals or children because it tastes sweet. So ethylene glycol tastes enough like a sugar, you know, the glycol part of it. So even like artificial sugars in gum, so sorbitol and glycol, you know, so you have kind of a crossover in your taste buds. So ethylene glycol tastes sweet enough that if you have a spill of antifreeze in your garage and a little kitty cat, The cat will drink it and then be like poisoned. Okay, and so the way that that's treated in the vet ER or in the emergency room is an infusion of ethanol. Okay, so not fun the next day, perhaps. So why does that work? It's a competitor. So that tells us that the methanol itself is not toxic, because then your kidneys will just flush out the methanol and not, so it's the methanol that's not bad, it's the byproduct. And so if you give your liver alcohol dehydrogenase enough other stuff to do, it will bind to the ethanol instead of the methanol or ethylene glycol, and those other molecules will just flush through. Okay, so I don't know that this ever actually happens, but there was an episode of ER one time where they gave like an intravenous drip of straight ethanol. So I would think you would have like the world's worst hangover the next day, but you would be alive. Okay, so that's a good thing.
I thought they used to put like that or something similar to it.
In ice cream?
Yeah, no, like a lot, like before they really like.
I don't know. That would be worth investigating. Ethylene glycol. Yeah, I don't know. Yes? Yes. Okay, so sometimes, individuals of Asian descent have issues with not enough alcohol dehydrogenase. Okay. Could be. So I had an Indonesian grad student who could drink like a fish. If he ever got pulled over, so a breathalyzer test would not show him up as drunk, because what they measure is the byproduct of ethanol metabolism. And so if you don't metabolize the ethanol, you don't appear, so if you did a blood alcohol test, it would show up as drunk, but not with a breathalyzer. So it doesn't get metabolized the same way.
Is it for other people too, though, like not just?
I mean, so I think that's an overgeneralization, just like, people 60 and over have lactase deficiency. So I think it's a common feature, but not exclusive. Absolutely.
I think I do.
You think
Not
you're
from
fine on that?
experience.
Not from experience. You've had genetic testing done, and so you say you think you're deficient, or you think you're not deficient?
I'm deficient.
Okay.
Not
Not from personal experience. I won't ask. Right. Okay. Any other questions about that? We're not going to get to gluconeogenesis today. What questions do you have on glycolysis? So does that still seem overwhelming? You're like, yeah, I've seen it a million times. We just need to refine it a little bit.
Can you go over which enzymes we need to know the mechanisms?
Which enzymes you need to know the mechanism for? Good question. Okay, so let's come back to our list of glycolysis enzymes. Okay, so you should know the mechanisms for. Okay, that's not I'm trying to erase it, but it's not erasing. Okay, I'll just circle them. Okay, so you should know the mechanism for boring, not hard. You should know the mechanism for phosphofructokinase 1, not hard. So isomerase, so we certainly did that mechanism. The one that we didn't do, but I said you should be able to do it as practice, triphosphate isomerase. Glyceraldehyde 3-phosphate dehydrogenase, and phosphoglycerate mutase. So certainly the other kinases that are just substrate level phosphorylation, you could certainly do those in your sleep. We didn't go over them. It's just kind of the other way, ADP grabbing that phosphate. So not a big deal, but we'll say that those, 6 are plenty. And then alcohol dehydrogenase and lactate dehydrogenase. But again, once you've seen one dehydrogenase, you've seen them all. It's really just knowing what the substrates and products are. Lactate dehydrogenase and ethanol dehydrogenase. Or alcohol dehydrogenase, rather. Okay, what other features of glycolysis might you want to tackle? So not just the mechanisms. So certainly substrates and products. Okay, so there's only 11, right? So that's not so hard. And so you already know glucose and fructose. So you just slap a couple phosphate groups on there. You know glucose 6-phosphate, you know fructose 6-phosphate. fructose 1,6 bisphosphate. Okay, so you're halfway there, right? What's that? So so don't learn the numbers, but you should know which ones are near equilibrium, which reactions are near equilibrium, and which ones are metabolically irreversible. You might want to know which reactions consume ATP and which ones produce ATP. You might want to know some features of them, so which ones use covalent catalysis.
Should we know, like, actually?
Carter, did you have a question?
No, it was answered.
Okay. So you can start working on that, right? Okay, other questions?
I think that would be biosignaling again.
Biosignaling will be on the exam. I will answer any questions on that on Tuesday. So I will not be here Thursday, but please come. So what Kang Meng will do, so Kang Meng is going to finish up biosignaling on Thursday. And then I'm going to give him a couple of practice problems to go over with you. So you can practice end of chapter problems, practice some delta G problems. You could ask him questions if you want. Might not be a full class period on Thursday. And then we'll pick up again on Tuesday. I'll answer any questions you have. We'll do gluconeogenesis, but I guess it can't be on the exam. Darn it. Okay, so the exam will just go through glycolysis. And, including signaling.
And that recording from last year covers what's going on Thursday.
Yes. So the recording from last year, I will have to look back at it and see. So if I said anything in that recording that wasn't covered at all in what Kang Meng said, so I don't want to force you to watch that recording. I want it to be helpful without giving you more burden. Okay. So have any of you tried to look at it? Is it helpful? Is it clarifying? Or is it just like, oh, there's way too much, there's twice as much information now?
Yeah, it was helpful.
Okay, so I want it to be helpful. I don't want you to feel obliged. Okay. I probably gave a quiz that time last year. Yeah. Okay, yeah, so the recording should be helpful, but if there's like a nitpicky little side story or tangent, that won't be on there. Just kind of the facts.
How do you click to play your recording? How do you click to play your recording? Is it not recording your voice?
It should be.
Okay.
Do you not have your volume up, maybe? When I'm recording or when you're playing it back?
Playing it back.
There should just be a YouTube video that shows up with volume. I think everybody else gets it. Thank you.
For the rough draft,